Hello everybody, I am Aarti Yadav, student of Department of Dairy Science and Food Technology, Banaras Hindu University. Today we are going to discuss about nutrigenomics. So these are the topics which we are going to go through in this presentation. So let's begin our presentation with a little background on this topic. So how did we come from nutrition to nutrigenomics? So researches on nutrition can be seen since 400 BC when Hippocrates speculated the hypothesis that the warm body temperature was innate. Also during 1700 AD, the so-called analytical chemistry era began. In the 19th century, Liebig identified carbohydrates, proteins, lipids and other macronutrients that released heat. In the 18th and 20th century, the era called chemical and analytical era of nutrition began. And in the 19th century, later, which was also called the biological era, the studies on metabolism and chemistry were done, helping the science of nutrition and defining their role in the development and prevention of chronic diseases such as cancer, neurodegenerative and bone metabolism disorders. So nowadays, the era which is going on is known as post-genomic era. This era is characterized by the integration of three fields: biological, social, and environmental, where scientific discoveries on nutritional pathophysiology and metabolism are included. The launch of the Human Genome Project in the 1990s and the subsequent mapping of the human DNA sequencing jump started the field of nutrigenomics that we know today. So what is actually nutrigenomics? Nutrigenomics is the study of molecular relationships between nutritional stimuli and the response of the genes. It is broadly defined as the relationship between nutrients, diet, and gene expression. It seeks to elucidate how the components of a particular diet may affect the expression of genes which may have increased its potential or which could be suppressed. Now coming on to how nutrigenomics is connected to other omics sciences. So after the Human Genome Project, discussions and actions began on a new biological era, that is the post-genomic era, which is currently going on. The evolution of bioinformatics provided advances in omics science research. This science uses biotechnology to isolate and characterize a greater number of biomolecules from the same group, such as DNA, RNA, proteins, or metabolites. Hence, after genomics, other omics sciences appeared as revolutionary tools such as proteomics, metabolomics, and transcriptomics. The studies on nutrigenomics are focused on the effect of the nutrients over the genome, proteome, and the metabolome. Moving on, so let's see how nutrient interacts with the gene. Nutrient-gene interaction can occur in three ways, that is direct interaction, epigenetic interaction, and genetic variations. In direct interaction, nutrients after interacting with a receptor behave as transcription factors that can bind to DNA and acutely induce gene expressions. In epigenetic interactions, nutrients can alter the structure of DNA so that gene expression is chronically altered, and it has sustained effects throughout the person's life, or it can persist across generations. Third is genetic variations. These commonly occur due to single nucleotide polymorphisms and can alter the expression or functionality of a gene. Direct interaction. A key mechanism of action involving nutrient-gene interaction is the ability and necessity of some nutrients to act as ligands and bind with transcription factors. Now, these transcription factors bind to specific DNA sequences in the promoter region of specific genes, thereby either enhancing or suppressing gene expression. The binding of a nutrient to a transcription factors will affect the ability of transcription factor to bind to the DNA. This is the diagrammatical representation of retinoids from carrots and fatty acids from salmon, in which they bind to specific genes via peroxisome proliferation activated receptor complex, resulting in, for example, reduction of fatty acid synthesis or increase in fatty acid oxidation. How do nutrients participate in gene expression? So, the pattern of gene expression, protein expression, and the metabolite production in response to a particular nutrient or nutritional regimes can be viewed as a dietary signature. Now, major or minor nutrients. Major nutrients consisting of carbohydrates, fatty acids, and amino acids, and minor ones of iron, vitamins. They act as dietary signals and are detected by cellular sensor systems that influence gene and protein expression and subsequently metabolite production. And in this manner, nutrients participate in the regulation of gene expression in response to nutritional changes. Now, the nutrients modulate the activity of transcription factors or the secretion of hormones that in turn interfere with the transcription factor. This is an example which shows how the carbohydrate-rich diet modifies the hepatic gene expression. Nutrigenomics and obesity. The researchers have indicated that not only environmental factors but also genetic aspects are related to health problems, including DCNTs and metabolic syndrome. Now, recent studies show that 80% of the difference observed in the body mass index of twins are related to genetic factors. As obesity causes a chronic process of inflammation, the use of nutrigenomics to modulate this manner is highly promising. Other reports demonstrated that some food contain anti-inflammatory bioactives such as caffeic acid, tyrosol, quercetin, and lycopene in tomatoes, guava, and watermelon. These molecules act inhibiting the expression of COX2 and INOS genes by reducing the translocation of the kappa B nuclear factor from the cytoplasm to the nucleus. There are several other ways where bioactive compounds from food can interfere on genes. One of the primary mechanisms for gene expression modulation is during transcription, where the synthesis of inflammatory mediators occurs and has a fundamental role on numerous chronic illnesses, including obesity. Hence, interleukin 1 is one of these mediators, which after activation stimulates the production of many other molecules during the inflammation cascade. This is the diagrammatical representation of gene expression in obesity. Nutrigenomics and cancer. So earlier studies showed that the deficiency of micronutrients such as folic acid, vitamin B12, B6, C and D, selenium, niacin, etc. can cause changes in to the DNA similar to what is seen after radiation, and furthermore they were demonstrated to be narrowly related to development of cancer. The molecules present in contaminated food can produce toxic metabolites that may interact with DNA, modifying its structure and inducing mutations. It is the case of aflatoxin B1, which forms on adding compound able to bind to the N7 position of guanine residue, generating a new product. This new molecule cleaves then the interaction between the one sugar and one nitrogenous base of a nucleotide, leading to the formation of an apiurinic site. The mutation can thus cause severe damages on the liver, including necrosis, cirrhosis, and carcinoma. During the metabolism of folate, the folic acid found in food sources absorbed by intestine and through many chemical processes of catabolism and synthesis. It is transformed into 5-methyl tetrahydrofolate. The chemical component is necessary for the synthesis of methionine, which in turn is used during the process of DNA mutation. Nevertheless, various minerals work as protectors against cancer development, that is selenium, prostacyclins, zinc. This is the diagrammatical representation of how the dietary mutagens can cause cancer by causing the methylation of the DNA. and activation of the oncogenes. Nutrigenomics and the type 2 diabetes. So, type 2 diabetes is a multifactorial pathogenesis that involves the interaction between genetics and environmental factors. Genomic studies show that there are 65 single nucleotide polymorphisms associated with the risk of developing type 2 diabetes. With the advances of genome sequencing and the decoding of the human genome, tests of the detection of the single nucleotide polymorphisms related to type 2 diabetes became available to the public. In these exams, the patient is able to know if there is a genetic predisposition to develop the disease or not. However, caution must be taken on applying this test for clinical practice because once the test has been done and the patients who have shown negative results may become carefree and may develop diabetes later on due to lack of concern. This is the diagrammatical representation of cardiovascular disease and nutrigenomics and how the leptotome, metabolome, and proteome affect the cardiovascular risk. Case study on fat. So there was a case study done in this. This study investigated the effect of short-term nutritional intervention on gene expression in adipose tissue from lean and overweight subjects. The gene expression profiles were measured after consumption of polyunsaturated fatty acids, PAS, conjugated linoleic acid, and MCT, and a controlled spread 40 grams of fat daily for 9 days. The adipose tissue gene expression profiles of lean and overweight subjects were different, mainly with respect to defense, response, and metabolism. So the results were there was lower expression of genes related to energy metabolism in lean subjects. The expression of inflammatory genes was downregulated, and the expression of lipid metabolism genes was upregulated in overweight subjects. Now, coming to some pros and cons of nutrigenomics, some of the pros are due to increasing global burden of nutrition related non communicable diseases, nutrigenomics could help to develop more sustainable approaches to encourage dietary change at a population level. The discovery of these interactions, that is the nutrient gene interactions, will aid the prescription of customized diets according to each individual's genotype. Thus, it will be possible to mitigate the symptoms of existing diseases or to prevent future illnesses, especially in the area of non-transmissible chronic diseases. Now, coming to some of the cons, the complex biology of human beings makes a mechanistic understanding of exactly how dietary bioactives react in our bodies difficult to elicit. How to define the optimal intake of individual nutrients for the maintenance of human cells in a genomically stable way remains largely unknown. Diverse genetic backgrounds further complicate the prediction of phenotype. It's some more susceptible to certain conditions than others. So, coming to the summary of this presentation, nutrigenomics was born due to the need to move from epidemiology and physiology to molecular biology and genetics. Currently, there are steps that needs to be considered in order for nutrigenomics to be applied. The genes, the gene-protein network, and the strategy towards the determination of nutrients, influences of gene-protein expression. Nutrigenomics in a single experiment can generate multiple responses, so it is very necessary to be innovative in the approaches of this area. Although much of the early hype around nutrigenomics has not yet come to fruition, the field remains nascent and fast moving, with the potential to lay the foundation of truly personalized nutrition approaches tailored to individual. The establishment of pan-national organizations such as European Nutrigenomics Organization called NUGO and the International Society for Nutrigenomics and Nutragenetics have further served to increase the infrastructure and international collaboration around nutrigenomics research. In bringing together the science of bioinformatics, nutrition, epidemiology, molecular biology, and genomics, much remains to be both discovered and determined. But future nutrigenomics research will no doubt provide future intriguing insights into both nutritional science and the human genome. Thank you so much.